Hi class. I'm here to give you the Chapter 4 Part 1 lecture. So we're first going to get started with looking at the assignment, which is right here. So after Chapter 4 Part 1, you will be able to answer number 1 and number 2 on your assignment. So describe the functions of each layer of connective tissue. We're going to talk about 3 different layers. They each have their own role. And then we're also going to talk about the components that are within each layer. Second, stretch-induced force deficit. How does static stretching influence strength and power performance? So we'll talk about different types of stretching, including static, different durations of static stretching, and how does that influence our strength or power performance right after. And then, what is the stretch-induced force deficit? So that's what we will be talking about. Okay. All right, so we are talking about skeletal muscle. If you've taken A&P 1, which I think that's a requirement for this class, you should have a little bit of knowledge about skeletal muscle physiology, and if you've taken A&P 2, you'll have more extensive knowledge, which is helpful. But what you see here is the muscle, and it's broken down into all of its individual compartments. So what we're first going to talk about, we're just going to start with the top, and then we're going to learn about each individual layer. So when you think of a muscle belly, which is said right here, I want you to think of the biceps muscle. So the biceps brachii, we're going to talk about that muscle a lot. So thinking about your biceps brachii, which is this muscle right here, when I say the muscle belly, I'm talking literally about that entire muscle. And we're going to talk about the layers that surround that muscle, and then we're going to go deeper and deeper and deeper inside the muscle. Okay. Learning objectives. This is for the entire chapter, but today we're just focusing on anatomy and physiology. You can go over all these learning objectives at the very end of the chapter just to make sure you have a good understanding of all of these topics. For part 1, we're doing structure of skeletal muscle, layers of connective tissue, and then the components of a sarcomere. Okay, so first is basic organization of skeletal muscle. Okay, this picture is very similar to the last picture. It just kind of shows you that we start with the entire skeletal muscle right here, and then it's broken down into these each individual compartments. So this is the order in which a muscle fiber is separated into, or a whole muscle is separated into. So we start with an entire muscle. We can say this muscle right here is the biceps brachii muscle. And then we break it down into smaller components. You see a fasciculus is a bundle of fibers. And so you pull this here, these are all muscle fibers. And then you look at this, it pulls out. So this is the muscle fiber. And the muscle fiber is made up of multiple myofibrils. And then within the myofibrils are myofilaments, which is your sarcomere. So you do have to know this order, but we'll talk about it. We'll continue to discuss this order throughout this part 1. What we're going to start with first is learning about connective tissue. So the epimysium is the first layer of connective tissue, followed by the perimysium, which surrounds the fascicles, and then the endomysium, which surrounds the muscle fiber. So we're going to get into the details of each of those layers, and remember, that's one of the questions on your assignment. Before we do that, let's just talk generally, what is the role of connective tissue? So CT, as you see up here at the top, stands for connective tissue. It stabilizes, supports components of skeletal muscle. So we see our muscle tissue kind of over here on this side. This right here is one entire muscle, like the biceps muscle. Each of these layers is going to support each of the components. You see here it surrounds the muscle at each organizational level. So each layer will act independently and contain specific functions that allow for movement and force production. And we're going to learn all about those specific functions on the next few slides. One question we want to ask is what happens when connective tissue is lost due to injury or exercise-induced damage? So exercise-induced damage or an injury, that's like micro tears. So like micro tears occur in the muscle fibers. And what happens when that occurs, we basically are going to see a reduction in strength or power-related characteristics. This is due to the fact that force is unable to be transferred optimally throughout the layers. So let's say we have damage to this endomysium layer right here. If we have damage to this layer, it may be hard for force to travel to the perimysium and then to the epimysium, which travels to the tendon and then to the bone to create movement. So if we lose connective tissue, which does happen all the time with exercise, that's okay because the muscle tissue can repair itself and it can heal over time. But in the instance that we do see damage to muscle tissue, we're going to also see decreases in muscle strength and power. Okay, so let's start with the first layer of connective tissue. So we know that connective tissue basically wraps the individual layers or compartments of the muscle. The epimysium is the first layer that we talk about, and it surrounds the entire muscle belly. So this right here, we can say this is the biceps muscle. It surrounds the entire muscle there. It's going to separate the muscle and protect the muscle from friction against other muscles or other tissue or bones. So epimysium may help prevent friction. So that's one role of it. It also ensures that force is transferred to the tendon. So we know that the tendon, it's an fibrous elastic tissue sheath that extends beyond the flesh of the muscle and blends to form that rope-like tendon that we know of. So our tendons connect to bones, and so the epimysium helps to transfer force through the tendon and to the bone. That's one of the major roles of the epimysium, is transferring force. Basically, it ensures that force is generated and transferred. This layer consists of elastic and collagen fibers, which allows for elasticity. So this layer is also very elastic, which is helpful anytime we contract a muscle, it's going to shorten, and anytime we stretch or lengthen a muscle, it's going to stretch out. So this layer is very elastic and allows for the stretching of muscle tissue. This layer can also get bunched up with knots and adhesions, and when we get a massage or foam roll or just self-rub out a muscle, we're actually rubbing out this top layer. So when you touch a muscle, like if you touch a muscle on your body right now, when you press down, you're feeling that top layer there. And then yeah, you can press harder and feel more of the muscle. But that massage and foam roll that we can do sometimes as a recovery method really does help to make sure that that layer, the epimysium, is rolled out, laid out, smoothed out, so there's not a lot of knots and adhesions, which also interfere with transfer of force. But yeah, that's our first layer that you can write about. So what you need to know really is it's the top outer layer, surrounds the entire muscle belly, prevents friction from the muscle rubbing up against other structures, and transfers force through the tendon. Next is the perimysium. So same picture here. The perimysium surrounds something called a fasciculus or fascicle, so containing a bundle of muscle fibers called fascicles. So that's what we see here. This bundle where my arrows is circling around, that is the fascicle, and it contains muscle fibers. So a bundle of fibers, which you see, is called a fascicle. So what's the main role of a perimysium? It groups muscle fibers into bundles. These bundles can be 10 muscle fibers or 100 muscle fibers, depending on muscle size. It's responsible for shaping and organizing the muscle fibers and transmitting force to the epimysium. It does receive a blood and nerve supply that helps to innervate and activate all of these muscle fibers here. Looking at this picture, we see microscopic image of the perimysium. So the perimysium is where the black is, and then inside here is all the muscle fibers, or the groups of fibers. This layer contains an intermuscular fat content that we refer to as marbling. Marbling, if you know anything about steak, if if you know anything about meat, they have something called marbling, and it's just like the fat that's interlaced. So our intermuscular fat is basically in the epimysium or the perimysium. The perimysium kind of surrounds all of that intermuscular fat. It also contains a collagen network of elastin fibers, which helps give the tissue support, flexibility, and elasticity. Advances in research for the perimysium basically say that the layers play a role in transmitting lateral forces. We have these perimysial junction plates that absorb and distribute force, reducing stress placed on the fascicles directly. So it helps to transmit lateral forces. Think of like doing a lateral lunge really quickly or doing a cutting motion. That's a lateral movement. And so advances in research have suggested that this layer helps to transmit lateral forces and prevent damage to directly injuring the muscle fibers. Pretty cool. So yeah, that's kind of what we know about the perimysium. Next is the endomysium. So the endomysium surrounds each individual muscle fiber. So this picture you see here, I added endomysium is going to surround just an individual fiber. It's going to bind the fiber to its neighbor. It also supports capillaries that supply each fiber. So each fiber is supplied with capillaries, like a capillary bed that exists inside the muscle. So each muscle fiber has its own blood supply. The endomysium lies over the muscle membrane, which is called the sarcolemma, which we'll learn about, and it provides an appropriate chemical environment for the exchange of sodium, potassium, and calcium. This is needed when an electrical signal passes over it. This is going to be essential for excitation and contraction of muscle fibers. So like I said, the endomysium lies over the sarcolemma, and basically provides a chemical environment for it. It also prevents the signal for muscle activation from spreading from one fiber to another. This helps to allow for fine motor control. So there's a difference between the force needed to write with a pencil, to write really fine print, and the force needed to throw a baseball. So think about that motor control and think about how we have to control how many muscle fibers are activated in order to control the amount of force that we need. So this allows the muscle to specifically control force generation, so fiber activation doesn't result in activation of neighboring fibers if we don't want to activate the neighboring fibers. This is necessary for precise motor movements that only require a few fibers, like writing with a pencil or taking notes. So basically it helps to match the force needed for a specific task. This layer also contains something called satellite cells. So satellite cells lie between the endomysium and the plasmalemma. So we haven't even learned about the plasmalemma or the sarcolemma yet, but we will, I think, on the next slide or two. But we'll get there. So we'll talk about what the plasmalemma is, but it's just another layer of connective tissue. So what's the role of a satellite cell? It's going to aid in repair and growth of muscle cells. Satellite cells are stem cells. If you've heard of a stem cell, it's basically a myonuclei cell, so muscle nucleus, and it helps to differentiate into whatever it wants to repair a certain issue or damage to muscle. And I know that sounds kind of crazy, sounds like a lot, but what you need to know about satellite cells is it's a stem cell that it hasn't differentiated yet. It doesn't have a role yet, but it can choose a role, and the satellite cells that exist in the muscle can help to repair and rebuild damaged muscle fibers, which is important with resistance training and endurance or aerobic training. Anytime we break down muscles, the satellite cells have to rebuild them. So we need satellite cells, so that's what exists at this layer. They exist in between endomysium and plasmalemma. All right, so usually I would ask this question in class. Each layer of connective tissue is vital for enhanced performance. How? And normally y'all would answer me. But just a few things I want to mention for you is it will provide for tendon structure. We know the epimysium creates the tendon, which allows for force to be transferred to the bone. It also helps to protect delicate contractile elements. So all those layers of tissue help to protect the delicate layers, delicate components of muscle. And it ensures that the force generated deep inside the muscle is transferred all the way through each layer to the tendon and the bone, which allows for movement. So yeah, that's what you need to know. All right, so let's kind of switch gears a little bit, and then we will come back to skeletal muscle physiology. But let's kind of switch just for a second, and we're going to talk about the stretch shortening cycle. So this cycle consists of eccentric and concentric actions. So I would assume that most of you know what those actions are, but if not, we can talk about it briefly. An eccentric action is the stretch of the muscle, so when a muscle lengthens, and the concentric action is when the muscle shortens. So anytime we run, think about running, look at this picture here. The muscle, the calf muscle, and most all lower body muscles during a jog, those muscles are going to they're going to stretch and they're going to shorten back and forth. It's a cycle. It's repeated over and over again with every single movement. And so the question I have here is, what happens to muscle power if an eccentric action, the stretch, is not followed by a rapid concentric action, a rapid shortening? So what's the best way to explain this? Basically, elastic force is generated when we have a stretch. So anytime we stretch, we're storing elastic energy. So think about going down into a squat really quickly. So as you go down, your lower body muscles are stretching. And as they're stretching, they're storing elastic energy. In order for us to be powerful, that elastic energy has to be released. So in order for it to be released, we have to follow the stretch by a rapid concentric shortening action. And so that shortening action allows us to release that elastic energy, and we're powerful. We produce force. We produce power. So basically, what happens if the eccentric action is not followed by concentric? Well, all of that elastic energy that's stored in the muscles, it's going to be lost inside the muscle as heat. I'm going to say it again. Remember the first law of thermodynamics, energy transformation? Well, the elastic energy is stored in the muscle. And when it's released with a concentric action, it would be released as mechanical energy. It's released as us moving, or potential energy. But if it's not released, it stays in the muscle and it's transferred into heat. So yeah, energy transformation. So basically, and then we can think about it as like a rubber band. That's another good way to think about it, is when you stretch a rubber band, when you pull it, it's going to stretch really far. Hold on. Whoops, let me do something. Never mind. When you stretch a rubber band, it's going to stretch. And then once you release it, it's going to snap back. Our muscles act very similar to a rubber band. But what happens when you stretch a rubber band too far and don't release it? Well, all of that energy isn't released and that rubber band stretches, and it doesn't recoil as quickly. And so that's kind of what we're about to talk about when we get into the stretch-induced force deficit on the next slide. So, okay, I would normally, again, turn it over to the class where y'all talk about these questions. So basically what you're supposed to answer: how does static stretching influence strength and power performance? Timing of stretch, duration of stretch. I hate this because this is one of my favorite parts, is when my students answer questions in class, because that means y'all are thinking and learning, and I'm sad we can't do that for this lecture. But I'll just kind of go over it. So we probably know what static stretching is. It's just like bending over and holding or touching your toes and holding that stretch. That would be a stretch of the hamstrings. So basically just bending over, touching your toes, holding that position. So that's static stretching. You can do that on any muscle group. How does that impair or influence performance? Well, an optimal static stretch needs to be less than 30 seconds per muscle area. And that's going to be beneficial for injury prevention and improved performance. We know dynamic stretches are also beneficial for this, but it needs to be about 30 seconds. And then duration of stretch, 30 seconds or less per muscle area. If we extend that to 60 to 90 seconds per muscle area, that's actually been shown to decrease strength and power performance and may increase injury risk because it decreases muscle stiffness, but it also increases compliance of the muscle. So you're probably thinking, well, isn't decreasing stiffness, isn't that a good thing? We don't want to be stiff. Well, there's a certain amount of stiffness that we need to perform well. And that's where we get into, and I say it again here. Let me fix this. It's not strength induced. It's stretch induced. Okay, sorry. Stretch induced force deficit is basically too much stretching. And too much stretching is going to inhibit neural activation of the muscle fibers. And that's going to decrease muscle fiber twitch force. And it's going to create elongation of the elastic compartments, which reduces the recoil force of the muscle. So it basically means we're stretching the muscle out too much. And when we stretch it out too much, the signal to activate the muscle fiber has to travel further. And so our power output is delayed, and we're not as snappy or as quick with our movements. So yeah, it's going to reduce our power capabilities of the elastic connective tissue, inhibiting explosive type performance. So what type of stretching is best before strength and power events? It's going to be dynamic stretching. Static stretching, like I said, is good if you keep it less than 30 seconds per muscle area or perform a dynamic workout. You would normally do the dynamic workout first before the static stretching. Dynamic stretches are like doing high knees or doing butt kicks back and forth, doing lunges down and back, doing what else? Bounds or backpedaling or karaoke or the grapevine thing or whatever. But it's any type of movement where you're moving your body dynamically through a large range of motion and not holding a pose. So that's going to be the best type of stretching. And then you can do all the static stretching you want, like after you get done with your strength or power sport or event. When you don't have to perform, stretching is great. So I'm not saying stretching is bad. I'm just saying you want to minimize the static stretching before explosive type movements because that's going to decrease your recoil force and decrease power output. Kind of interesting. So yeah, now you have all the information you need to answer number 2 on your assignment, number 1 and number 2. So yeah, if you have a question about this topic, you can reach out to me and we can go over it further. I know a lot about this topic because my husband, Dr. Palmer, does all of his research in stretching. So I have heard every single thing about stretching that a person can hear. So please let me know if you have a stretching question. I can answer it for you. All right, so now let's get back to where I said we were going to talk about the plasma lemma and the sarcolemma. So plasma lemma, as you can see, you would assume a plasma membrane. It surrounds each muscle fiber. So remember when I said that satellite cells exist between the endomysium and the plasma lemma? So basically the plasma lemma surrounds the muscle fiber and then the endomysium is right above the plasma lemma. It helps in transmission of signals. Remember I said earlier it's going to create a nice chemical environment, nice chemical environment for the exchange of sodium and potassium and calcium. Also helps to maintain the acid-base balance and transport metabolites. So this is where that acid possibly could accumulate when we have during exercise. When we're exercising, the acid can escape the muscle cell and it can accumulate in different parts of the muscle, and one part is the plasma lemma. But this part of the muscle helps to maintain that acid-base balance, helping to kind of minimize acidity. And then we have the sarcolemma, which is composed of the plasma lemma and a basement membrane. So sarcolemma is made up of the sarcoplasm. Y'all, we've talked about the sarcoplasm and the cytoplasm. If you hear all that noise, those are my dogs playing. I'm so sorry about that. Okay, it's made up of, so the sarcoplasm we have talked about, and it's like the cytoplasm. It's a gelatin fluid-like substance. So fluid that fills the space between the myofibrils, contains proteins, minerals, fats, glycogen. But yeah, so I will not be asking a ton of questions about the difference between plasma membrane and plasmalemma and sarcolemma. I really won't. I may just ask a very simple question about the two. So just have a decent understanding of them, but I'm not going to ask you like the difference really between the two of them because they are very similar and they're very close together. All right, another structure we need to talk about, or two more, is one is transverse tubules. These are little, like it says, tubes that pass laterally through the muscle fiber and transmit nervous impulses, which is an action potential. So it passes through the muscle fiber, transmitting the signals. We'll look at a picture later and it'll show these little, they're kind of like invaginations inside the muscle. So these are just little tunnels. And then another structure we're going to learn about is the sarcoplasmic reticulum. Sarcoplasmic reticulum is a network of those transverse tubules that loop around the myofibrils and they store calcium. So calcium, we're going to talk about, is important for muscle contraction, and we need it to contract muscles. And where is it stored? It's stored in the sarcoplasmic reticulum, which is, again, it's just a network of tubes that surround the myofibrils containing calcium. All right, so continuing kind of with what we're talking about, let's talk about, we just talked about here, we talked about myofibrils, so right here, now let's talk about muscle fibers contain thousands of myofibrils. So this right here you can see is a muscle fiber, and then you see all these little, they kind of look green or gray, all those little dots, those are myofibrils. So depending on muscle size and location, it could contain 100 or thousands of these little myofibrils. So what is the role or purpose of the myofibril? The myofibril houses all of these sarcomeres, and so that's what you see, the red and the green, you see kind of striated through here, that is a sarcomere, and that contains myofilaments. The filaments are actin and myosin, which we are about to learn about. So this here is a sarcomere viewed through polarized light. So, and what we see here is striated banding pattern. So we see a relaxed muscle, just means that you're not flexing it. You're not flexing your muscle, it's relaxed. This is what your muscle would look like under polarized light. So you see no overlap, you see bands, you see dark areas and you see light areas. Well, the dark areas are the myosin and the light areas are the actin. And then we see down here, we see more of this striated pattern. We see this overlap. So the arrangement of these filaments give a striated appearance, which is the overlap of the two. You see here, this is, fully contracted. So it is a fully contracted muscle fiber, and when we contract, we have this striated pattern. The striation, which is a striped appearance, is due to the overlap or arrangement of the myofilaments, actin and myosin, when a muscle contracts. Give me just a second. Okay, let's see. Let's get back to, okay, so now we're going to talk about those myofilaments. So actin is the first one. Actin we see here is, it's depicted in this picture as these yellow little ball-like structures. But what it is, it's an intertwined helix of actin molecules. They're basically just actin molecules, and then they have an active site. So active site is where you see kind of like the little black dots on the yellow. That's an active site. That's where the myosin head, the cross bridge, is going to bind to it. So yeah, that's what the actin filament is. And we have to talk about these two other structures. So the actin filament is wrapped in something called tropomyosin. So tropomyosin is the long chain that covers the active sites. So why does it cover it? Because it prevents myosin heads from binding onto it. These are called regulatory proteins because they regulate contraction. So they cover the active sites. That's what the tropomyosin does. And it's going to shift back and forth depending on if there is calcium present. And we'll talk about that. And then we have troponin. Troponin, we see, is the blue structures, and we have different subunits. We have I, C, and T, which we'll talk about on the next slide. So what you need to know is actin is a contractile protein, and it allows for contraction. It's the thinner filament. And then tropomyosin and troponin are regulatory proteins, and they will prevent contraction, or they regulate when contraction happens. Okay, so here are the different subunits. So troponin I is one subunit, and let me back up. In this picture, actin is depicted as the red molecule. So we have red actin molecules on this slide, and we have tropomyosin is this yellow and green twisted helix that is covering the active sites. So now let's talk about troponin. So troponin I binds to actin. So you can think I, actin. Hold on a second. My dog has just escaped something. Hold on. Sorry, Finn just took his food bowl out into the snow. Okay, so troponin I, think of I as an actin. That's what you want to think. It holds the tropomyosin complex to the actin. So that's the only role of troponin I. Troponin C binds calcium. So that one's easy. It binds calcium, which is the stimulus for muscle fiber contraction, and we will talk about that later, the main role of calcium and all that. But troponin C binds to calcium. Troponin T has an affinity for tropomyosin. Tropomyosin, we already know what this one is. It's this long helix of a protein that prevents contraction. So troponin T holds on to the tropomyosin. So yes, you do have to know each one of these components. You do have to know the difference between troponin I binds to actin, troponin C binds to calcium, and troponin T binds to tropomyosin. That's all you have to know for these three. And remember, they are regulatory proteins that regulate when contraction can occur. Okay, next is the myosin filament. The myosin filament is also a contractile protein, just like actin. So actin and myosin work together. Those are the myofilaments. So myosin consists of this globular head, which we see right here, a fibrous tail, and then a hinged pivot. All right, so the globular head is made up of an enzyme. Do you remember when we talked about the ATP synthase complex in the electron transport chain and that ATPase enzyme or ATP synthase? Well, I said there are multiple types of ATPase, and so here's another one. We also had an ATPase that we talked about hydrolyzes the ATP or puts it back together. That was the enzyme that we talked about. Well, when we add myosin to it, it's going to be kind of specific to the myosin head. So this head is made up of an enzyme, ATPase. ATPase splits apart an ATP, releasing energy. And so we need to know that for part 2 when we talk about the sliding filament theory. We're going to talk about how does the myosin head, the ATPase enzyme, how does it hydrolyze ATP. The fibrous tail will intertwine to form this long, kind of looks like a braided-like helix. So it's going to form that long part there. And the hinged pivot, I don't know if I have anything, allows for the head to basically pivot back and forth. These heads, so we have 2 myosin heads that are going to interact with actin, and when that happens, that's called a cross bridge. So the head groups of the myosin molecules form a cross bridge, so 2 of them. So 2 heads put together, which we see right here, this is a cross bridge. These heads are going to connect to the actin, the active site. Remember we learned about the active site? So the active site of the actin is where this myosin head is going to attach onto. And it's going to pull and it's going to create force. Okay, anything else on this slide? That's basically it. You just have to know myosin is the largest protein in the muscle. It's a contractile filament. It kind of looks like a golf club. That's what you can think about. And then when you have 2 of them together, 2 myosin heads, that's going to create a cross bridge. This is also called our thick filament. These heads are going to pivot back and forth, and they pivot back and forth, and they are going to connect to the actin. Okay, two more proteins we need to discuss is a non-contractile protein, also called a structural protein. So remember, this is the third type of protein that we've learned about. We had regulatory proteins, contractile proteins, and structural proteins. So these are structure, obviously, they provide structure for the positioning of actin and myosin, contribute to the elastic components of a muscle. First one is nebulin. Nebulin extends from the Z line and is localized to the I band. We haven't learned about the I band or the Z line yet, so don't worry, we will talk about it. But you can see in the picture right here, this is the Z line. It's like a boundary. It's like the end of one sarcomere is basically what a Z line is. So what does nebulin do? It mainly just works with actin. So it resides on top of actin and regulates thin filament length during sarcomere assembly, which is when our muscles are actually forming. Research suggests it does have a role in cell signaling and calcium homeostasis in the cell, but you don't have to know that. But you have to know that nebulin is a structural protein that stabilizes actin to the Z line, which is the boundary or the end of a sarcomere. And then down here we have titin. Titin is the third largest structural protein, third largest protein. Largest one's myosin, followed by actin, and then titin is third. It connects the Z line, which we see as the boundary, to the M line. The M line is the middle of the sarcomere. It's identical on either side, it's just flipped, okay? So the M line is the middle, the Z line is the end of it. So titin, what does it do? It functions as a molecular spring, and it stabilizes myosin. So I'll just put all this up there. Okay, so, and that's what I would normally ask in class. I would turn to y'all and be like, what does this yellow structure look like? It looks like a spring, and that's how it acts. It's a molecular spring responsible for passive elasticity of a muscle. Passive, not active. Active is like if I actively contract my muscle. Passive is if I just go down into a stretch or if I touch my toes. So if I go down and touch my toes, stretching my hamstrings, what happens here? Well, titin will respond, and it basically is going to limit range of motion. So that tightness that you feel, normally I would also do this. I would have all of y'all stand up and touch your toes, and you feel tightness. You keep your knees straight and you touch your toes. You feel that tightness there and a limited range of motion. One reason is because of titin. It contributes to passive stiffness. So preventing overstretching of the muscle is what it's doing. It also adds support and integrity. So those are our two structural proteins. Remember, they are not involved in the contractile process of muscle, but they hold the contractile proteins in place so they can form optimal actomyosin complexes, which create force. Okay, so now we're going to learn about how these components are kind of maybe just like different components of a sarcomere. So these components were investigated and proposed in 1954. These were ways for researchers to just identify different parts of the muscle. So the first one we're going to talk about is the A-band. The A-band extends the entire length of the myosin filament. So wherever the myosin is, in this picture, the myosin is red. So wherever the myosin is, that's going to be the A-band. The A-band overlaps the actin. So you see there that there's some actin in this gray box, but we have all of the myosin and the M line. So the M line and all of the myosin is within the A-band. Investigators have noticed that the A-band remains relatively constant in length during a contraction. So when we contract, the A-band actually does not change. It does not move and it does not shorten. One thing I didn't mention, but we can talk about now, is when you shorten a muscle, the myosin filament actually doesn't move at all. So think about your bicep muscle again. Think about moving your elbow. So bending your elbow, you're moving that biceps muscle, but the myosin filament is actually staying the same. It's stabilized to the M line. It does not move. So that's your A-band. Next are the I-bands. So the I-bands are at the edges of the sarcomere, and they only contain actin. So they only contain the actin, which we know is the lighter, less dense area of the filament, and there's no thick filament inside the I-bands. Investigators have noticed that the I-bands change its length along with the sarcomere. So our sarcomere will actually shorten with contraction, and we'll look at that next time, or in part 2. We'll look at a lot of videos about how does the sarcomere shorten. So it's going to shorten, and when the sarcomere shortens, the I-bands will also shorten because actin is tethered to structures located at the lateral ends of each sarcomere called those Z-line, or in this picture, a Z-disc. So these are a Z-disc. This is tightened here. Nebulin is not shown, but you can see that actin is tethered to the ends. So when the sarcomere shortens, that's going to create shrinking of the I-bands. How does the sarcomere shorten? Well, if you look here, you see the myosin heads are attaching to the actin. When it attaches to the actin, it's actually going to pull it towards the M-line. That's going to decrease the strength, or not the strength, the size or the length of the I-band, and it decreases the actin on the outside. Next is the H-zone. The H-zone is in the middle of the sarcomere, and it contains only myosin. You're like, wait a second, wasn't that the same as the A-band? The A-band contained all of the myosin filament. This contains only, the H-zone is only the myosin without the actin. When a sarcomere shortens and contracts, the H-zone will actually shorten as well. It's going to shorten and disappear, actually. Why? It's due to the actin being pulled towards the center of the myosin. It's actually going to slide in there. So think about your bicep again. So stretch out your elbow, or stretch out your arm, and then contract your elbow, your bicep shortens. So when the bicep shortens, these thin filaments, your actin, is going to be pulled toward the M-line. That shortens the entire sarcomere, and this H-zone is going to shrink because now the actin slides into it. So it's going to shrink and disappear because the actin is going to slide all the way into, kind of through the middle of it. And then lastly, which we've already kind of been over, is the middle of the sarcomere we said is called the M-line. It anchors myosin in place. And then the Z-lines, or the Z-disc, are going to define the end of the sarcomere, and they are going to connect to actin. So the M-line connects to myosin in the middle. The Z-lines connect to actin on the outside. So the M-line does not move. So it does not change in length when we contract a muscle. The M-line stays the same, doesn't move places, but the Z-lines do. The Z-lines are going to be pulled toward the M-line as actin is pulled toward the M-line. All right, so let's review. Shortening of a sarcomere. So this picture here kind of shows every zone and every band. As the sarcomere shortens, actin slides over the myosin towards the M-line. We already know the A-band does not change in length. It stays the same. The H-zone disappears because the I-band slide into it, and the I-band shorten because there's less of the actin on the outside. It has traveled towards the inside. And then as the sarcomere relaxes, actin returns to its original position. Again, the A-band never changed at all. The H-zone and the I-bands return to their original lengths. So that's the end of this lecture, but let's do a quick review and check out something really quickly. Okay, we're going to watch this video. You know me by now. I show lots of corny videos, but I really do think they help.
Skeletal muscles are attached to bones by tendons. Each skeletal muscle in your body is made up of bundles of muscle fibers. Muscle fibers are specialized cells. They contain multiple nuclei and are made up of myofibrils. The stripy appearance of the fibers results from the regular arrangement of the myofilaments within the myofibrils. In the myofibril arrangement, the sarcomere is one of the segments into which a fibril of muscle is divided. The M-line provides an attachment for myosin filaments. The Z-line provides an attachment for myosin filaments.
Y'all, I'm so sorry.
The A-band is the darker band of the myofibril containing the myosin filaments. The H-band is the lighter section in the middle of the A-band, where only myosin is present. The I-band is the lighter band of the myofibril containing only the actin filaments. When the muscle is relaxed, the protein tropomyosin blocks the myosin-binding site of actin. Muscle contraction occurs when the muscle receives an impulse from a nerve cell in the form of an action potential.
Okay, sorry about the dogs. But we finished the lecture. I did not finish that video, but you have that in your slides so you can watch that video. It goes over those bands. It goes over all the components. It will talk about stuff that we haven't talked about yet that we will talk about in the other parts. So thank you for watching and taking notes, but you can always rewatch this video over and over again. But yeah, so that is part 1 of chapter 4. If you have any questions over anything, please reach out to me. But yeah, that's it. Hopefully I will see y'all back in person very soon. Okay, bye-bye.